Hello, everyone. So, like Tom and Amanda said, my name is Leah Adams. I'm a rising sophomore. I have a biology major, chemistry minor. I am doing research under Dr. Tom Idy from the Patuxent Environmental Aquatic Research Laboratory. My presentation is about modeling PFAS biomagnification in the Chesapeake Bay. And I'm so excited to present today. So I hope you guys enjoy my presentation. Um, so PFAS are per and polyfluoroalkyl substances, and they're a family of organic chemicals that are produced all over the world and have been being produced for more than half a century. The characteristics that make them so useful is that they can resist heat, water, degradation, and many other chemical changes. However, this is also what makes them so strong as pollutants. Um, Today, they are found in firefighting foams used on Navy bases, as well as many of your household items, as you can see in the picture to my right. To name a few, pesticides, shampoo, nonstick cookware, and any stain-resistant products. So PFAS are made up of carbon backbones of varying lengths, and this is what separates them into their two groups of long-chain PFAS and short-chain PFAS. Long-chain PFAS, for the most part, are any PFAS with a carbon chain longer than 7 carbons, and short chain are any PFAS with 7 or less carbons, with a few exceptions. Long chain PFAS, in comparison to short chain PFAS, are more hazardous, they're more persistent, and they bioaccumulate in a food chain to a higher degree. Long chain PFAS, PFOA, and PFOS are the most abundant in the environment right now, and they're the ones that people have done the most research on. PFOA is the main concern for drinking water, and PFOS is the main concern for bioaccumulation throughout food chains. So in our study, we expect to test for some 20 PFAS, but based on the information that we have already found, we only really expect to see PFOA and PFOS in an abundance. So PFAS are a cause for concern because they are extremely mobile, persistent in the environment, and they bioaccumulate. If you look at the diagram to your left, labeled common PFAS exposure pathways, you can see that PFAS can travel through many different pathways, ultimately leading through human exposure. So we can be exposed through consumer goods, such as fire retardant clothes, nonstick cookware, and Scotchgard, among many other things. And another pathway to human exposure is through contaminated food, and more specifically contaminated seafood, and that's what our study is going to be on. Organisms such as the Chesapeake Bay blue crab can live in PFAS-contaminated waters, often from firefighting foams or industries that have leaked the contaminants into their water. And when we eat them, we are exposed to whatever they were exposed to. So on the on the other side, if you look at the image to my right, you can see some of the health effects that PFAS exposure has proven to cause. To name a few, PFAS exposure may cause low infant birth weights, decreased fertility in women, high cholesterol in both men and women, and decreased vaccine response in both. So the question that we're trying to answer through this research is, can the Chesapeake Atlantis model, the model to our left, can it be used to predict PFAS contamination in the Chesapeake Bay? So the colors, as you can see, represent changes in salinity. And every little box in the model is another area that will get a predicted PFAS concentration. So ecosystem models are key for understanding all the working parts of an ecosystem and being able to evaluate the influences that different changes may have on the system as a whole. So the model that we'll be using is parts of the Atlantis model, hence the name Chesapeake Atlantis model. And some of the things that will be integrated into our model are predator-prey relationships, nutrient cycling, biomagnification of PFAS, of course, and more. If you look to the right, there are about 46 groups. Well, there are about 46 groups that we'll be studying, but the pictures on the right represent only a few of them. And these will be the organisms that we'll be testing, that we'll be looking for information from as far as PFAS concentrations and biomagnification factors and transfer rates of PFAS. So the first step of this project is doing a literature and data review to feed the model. This is where I've been spending most of my time. And the purpose of this is to find PFAS concentrations in our target organisms, biomagnification factors, and transfer rates of PFAS, so we can put all that into the model. We've also been looking for spatial concentrations of contaminated water, possible sources of PFAS, and the uptake rate of all organisms in the model. Our second step will be collecting the blue crab from areas that we predict will have low or high levels of contamination, and then bringing it to the laboratory to test the contaminants in the muscle tissue to see whether or not our model predictions are valid or invalid. So as of right now, I have been able to gather concentrations for the full organisms as well as biomagnification rates and transfer rates. We have gathered information from both human studies and animal studies that model humans that have clearly documented the negative effects that PFAS can have on human health. As of right now, there are many states that have state fish advisories, including Michigan, and these are just in place to let you know which fish you can eat, which you shouldn't eat at all, and how much of them you should be eating. As of right now, Maryland is in the process of putting more regulations in place. However, starting on October 1st, there will be a new law that prohibits the use of firefighting foams that intentionally contain PFAS chemicals. In a study done by the US Navy, they found more than 240,000 parts per trillion of PFOS and PFOA in groundwater. Let's just say that is a lot. That's significantly above the amount that I guess different places have said is the safe guideline. So this is proof that there needs to be more regulations in place and finding a way to get rid of all these chemicals. So as I said, the research is only in its preliminary stages, so there aren't any conclusions that we have drawn yet. However, the long-term outcomes will be the ability to evaluate the threat that PFAS pose to humans and harvested species from the ecosystem, as well as a possible application of the model for other harmful substances that may plague the ecosystem. And I would like to acknowledge the NIH RISE staff for funding my research and helping me to become a better researcher and presenter, as well as Dr. Tom Ivey. Thank you.